Nothing about the Mariana Trench is normal. Not the depth, not the pressure, not the life that clings to its walls against what should be impossible odds, and not what humanity has managed to deposit at the very bottom of it. Here's why. Start with geography. The Mariana Trench occupies a narrow corridor of the western Pacific Ocean, roughly 200 kilometers east of a chain of volcanic islands that bears its name. The Mariana Islands, themselves named after Queen Mariana of Austria, the 17th century queen consort of Spain, form the visible arc above one of the most extreme geological events happening on this planet right now. The Pacific Plate, some of the oldest and densest oceanic crust on Earth, up to 170 million years old, is being forced beneath the smaller Mariana Plate to the west. Old, cold crust is denser than young crust. It sinks, and where it disappears is the Mariana Trench. The deepest result of that disappearance has a name: Challenger Deep. The trench runs approximately 2,550 kilometers in length, 69 kilometers wide at its broadest point. Modern high-precision sonar surveys, corrected with conductivity, temperature, and depth data from the water column, put Challenger Deep at somewhere between 10,925 and 10,935 meters below the ocean surface. Call it 10.9 kilometers. Call it nearly 7 miles. Call it the deepest point on the surface of the planet. None of those numbers communicate scale to a human brain, so try this. Mount Everest stands 8,849 meters above sea level, the highest point on land. Turn it upside down and drop it into Challenger Deep, and the former summit would still be underwater, more than 2 kilometers short of the trench floor. Nearly 7,000 feet of open water above where the peak would hang. The trench swallows the biggest thing on land and has room to spare. And almost no human being has ever been there. On January 23, 1960, U.S. Navy Lieutenant Don Walsh and Swiss oceanographer Jacques Piccard descended in the bathyscaphe Trieste, a pressurized sphere barely 6 feet in diameter, attached beneath a float of aviation gasoline. Walsh was 28 years old. As the Trieste neared the bottom, an explosion rocked the craft. They couldn't identify the source, and they kept going. They reached approximately 10,916 meters, spent about 20 minutes at the bottom, observing a seafloor of pale diatomaceous ooze, and then ascended. Nearly 5 hours to the bottom, another 5 hours back. Then 52 years passed. It wasn't until March 2012 that a second crewed vehicle reached Challenger Deep. Filmmaker James Cameron, who spent years and tens of millions of dollars on the project, descended solo in the Deep-Sea Challenger, a torpedo-shaped submersible designed specifically for the dive, to approximately 10,908 meters. That brought the total number of humans to reach Challenger Deep to 3. 12 human beings had walked on the moon by that point. Fewer people had seen the deepest point on Earth than had left boot prints on another world. Regular visits only became possible in 2019, when explorer Victor Vescovo commissioned the DSV limiting factor, the first commercially certified full ocean depth submersible. Between April 28th and May 5th of that year, Vescovo and his team made 4 dives to the bottom of Challenger Deep, more visits in 8 days than in the previous 59 years combined. On April 28th, Vescovo reached 10,928 meters, a new depth record. Don Walsh, who had first reached that floor in 1960, was on board the support ship when Vescovo surfaced. As of July 2022, 27 people had descended to Challenger Deep. The trench has grown fractionally more accessible, but the number still defines a kind of inaccessibility that almost nowhere else on Earth can claim. Pressure is the reason. At Challenger Deep, the water column above exerts a force of approximately 1,086 bar, roughly 15,750 pounds per square inch, about 1,072 times the atmospheric pressure at sea level.
One account of deep-sea research describes it as pressure that is thick, extremely heavy, more than 16,000 psi, or 1,100 times our own atmosphere. At the surface, The atmosphere presses on your skin at about 14.7 pounds per square inch. At Challenger Deep, that number exceeds 15,000. An unprotected human body would compress and collapse before reaching a fraction of that depth. This isn't a place where adaptation is an option for familiar biology. It's a place where familiar biology simply does not apply. Temperature at the bottom holds between 1 and 4 degrees Celsius. A 1996 in situ measurement recorded 2.6 degrees at 10,897 meters. Not frozen, the salinity suppresses the freezing point, but cold enough that most biochemical reactions proceed at a crawl. And there is no light. Not dim light, not filtered green twilight. No photons reach Challenger Deep. Sunlight penetrates to roughly 200 meters in clear ocean water and fails entirely by 1,000 meters. Challenger Deep is nearly 11,000 meters down. The darkness is absolute, and has been absolute for the entire geological history of the trench. Without light, there is no photosynthesis. Without photosynthesis, there is no food web rooted in plants or algae. Everything alive in the hadal zone ultimately depends on material that has sunk from the sunlit world kilometers above. Dead organisms, fecal pellets, organic particles drifting down in a perpetual, slow rain called marine snow. What makes the trench unusual is that its V-shaped geometry funnels this material toward the deepest axis, concentrating nutrition in a way that the broad, flat, abyssal plains can't replicate. The bottom of the deepest trench on Earth is, counterintuitively, not a food desert. It's a depositional sink, and what gets deposited there isn't only organic. To understand what occupies this zone, you need to understand how the ocean is divided vertically. From the surface to 200 meters is the epipelagic zone, the sunlit zone, where photosynthesis happens and most of the ocean's familiar biodiversity concentrates. From 200 to 1,000 meters, the mesopelagic or twilight zone. Some residual light filters into the upper portion, but not enough for plants. Bioluminescence becomes common here, organisms producing their own light to hunt, communicate, and hide. Below 1,000 meters, the bathypelagic zone, the midnight zone, extends to 4,000 meters. Complete darkness. Extreme pressure by surface standards. Then the abyssal zone from 4,000 to 6,000 meters, where the ocean floor levels into broad plains and life thins dramatically. And then, at 6,000 meters, the hadal pelagic zone begins. This is the only oceanic zone that exists almost exclusively inside deep ocean trenches. Every zone above it, epipelagic, mesopelagic, bathypelagic, abyssal, extends continuously across the open ocean. The hadal zone does not. A few oceanic depressions outside of trenches technically qualify, but they're exceptional cases. The hadal zone is a phenomenon of subduction, confined to narrow geological slots like the Mariana Trench, the Kermadec Trench, the Tonga Trench. It occupies less than 0.25% of the world's seafloor by area, yet spans roughly 40 to 45% of the ocean's total vertical depth range. That fraction of the seafloor contains more than 2/5 of the ocean's entire depth. The zone that barely registers on a map accounts for an outsized portion of the planet's interior volume. Inside it, life exists. Not abundantly, not in the obvious vertebrate form, but it exists, and some of what survives there is genuinely alien in appearance. The most remarkable vertebrate resident is Pseudoliparis swiri, the Mariana snailfish. Formerly described as a new species in 2017 by marine biologist Mackenzie Geringer and colleagues, it was collected at 7,949 meters in baited traps. The fish is small, translucent, and gelatinous. Its skeleton is poorly mineralized. Its skin is thin enough to look through. It feeds primarily on amphipods. Japanese scientific teams from JAMSTEC and NHK captured video of P. swiri at 8,178 meters on May 18, 2017. The depth record for this species in the Mariana Trench specifically, and a figure that sits close to what the chemistry of pressure predicts as an absolute ceiling for fish. More on that mechanism in a moment, because it's the most important biological fact about this place. Deeper still, where no fish can function, single-celled organisms called xenophyophores persist in the sediment. Xenophyophores are foraminifera, giant protists, and individual specimens can measure up to 25 centimeters across. They are single cells, one cell large enough to cup in two hands, surviving at approximately 10.6 kilometers depth in the Serena Deep. They are among the deepest living known eukaryotes on the planet, able to withstand elevated concentrations of heavy metals and function in conditions that would destroy anything more biochemically complex. In the midnight zone above the hadal threshold, the giant phantom jellyfish, Stygiomedusa gigantea, drifts with a bell exceeding a meter in diameter and trailing arms up to 10 meters long. It's been recorded as deep as 6,665 meters, the very edge of the hadal zone, and it's dark red, a color that disappears in deep water, making it effectively invisible to predators operating in near total darkness. The giant isopods of the genus Bathynomus, reaching 50 centimeters in length, occupy the abyssal zone at depths up to around 2,500 meters, well above the hadal threshold. They're not hadal creatures. The distinction matters enormously because what the hadal zone does to body size runs against the expectation that deep implies big. The reason for that counterintuition lives inside a molecule, trimethylamine N-oxide, TMAO. In shallow water, TMAO is mostly known for its consequence. When bacteria convert it to trimethylamine after a fish dies, you get the characteristic smell of rotting seafood. In living deep-sea fish, it serves an entirely different function, and understanding that function requires understanding what high pressure does to biology. Hydrostatic pressure, the weight of the water column above, increases by roughly 1 atmosphere for every 10 meters of depth. At 10,900 meters, the pressure is more than 1,000 atmospheres. That pressure does something specific to proteins. It disrupts their three-dimensional folding. Proteins aren't simple chains. Their function depends on their shape. Enzymes have active sites that only work in precise configurations. Structural proteins hold cells together in specific architectures. Membrane proteins open and close ion channels at exactly calibrated geometries. Pressure compresses water molecules around proteins and perturbs the hydrophobic interactions that hold folds in place. The protein unfolds. The enzyme stops working. The cellular machinery fails. TMAO counteracts this. Marine biologist Paul Yancey at Whitman College has studied this mechanism for decades. His landmark 2014 paper in the Proceedings of the National Academy of Sciences, Marine fish may be biochemically constrained from inhabiting the deepest ocean depths, established the formal quantitative relationship. TMAO acts as what researchers call a piezolyte, a pressure-counteracting solute. It works by strengthening hydrogen bonds in the water surrounding proteins, functioning as what one study describes as a structural anchor for water under pressure. The molecule acts as a molecular brace, holding proteins in their functional shape against the force trying to flatten them. One accessible summary of the mechanism puts it simply: TMAO's water-managing ability protects a critter's critical proteins, including muscle, from crushing under deep ocean pressures. The data from Yancey's study made the depth dependence stark. Shallow water teleosts carry roughly 40 to 50 millimoles of TMAO per kilogram of tissue. At intermediate depths around 1.4 kilometers, the concentration begins scaling approximately linearly upward. By 2,900 to 4,850 meters, abyssal grenadier fish carry 173 to 261 millimoles per kilogram. A hadal snailfish from the Kermadec Trench, living at approximately 7,000 meters, carries 386 millimoles per kilogram, the highest recorded TMAO concentration in any fish tissue. The more depth, the more TMAO required to keep proteins functional. This relationship holds across species that aren't closely related, which suggests it's not an evolutionary preference, but a physical constraint that convergent evolution keeps rediscovering. Here is where the chemistry stops being impressive and becomes lethal. Bony fish, teleosts, the group that includes virtually all familiar fish species, are hypoosmotic regulators. Their internal body fluids are considerably less concentrated than seawater. A typical shallow water teleost maintains an internal osmolality of around 350 milliosmoles per kilogram of fluid. Seawater runs at approximately 1,100 milliosmoles per kilogram. The fish is saltier outside than inside, which means water continuously tries to leave its body through osmosis. The fish counteracts this constantly, pumping ions out through specialized cells in its gills, producing concentrated urine through its kidneys, spending significant metabolic energy just to maintain the gradient that keeps it from dehydrating into the ocean around it. TMAO is an osmolyte, which means it contributes to the internal concentration of the fish's body fluids. Every millimole of TMAO a fish accumulates to protect its proteins also raises its internal osmolality. The Kermadec hadal snailfish at 7,000 meters already showed an internal osmolality of 991 milliosmoles per kilogram, almost at seawater concentration. Yancey's data, extrapolated across the observed depth TMAO relationship, predict that a teleost at approximately 8,200 meters would have accumulated enough TMAO to reach an isosmotic state, its internal fluid concentration equaling that of the surrounding ocean. Subsequent analyses, including updates Yancey published in 2025, place this threshold at 8,200 to 8,400 meters. At that point, the fish faces a paradox with no chemical exit. If a fish somehow survived to just below that threshold, its internal osmolality would begin to exceed seawater. It would become hyperosmotic, internally more concentrated than the ocean surrounding it. Water would begin flowing into the fish from outside rather than out. The gills and kidneys that evolved over hundreds of millions of years to handle the hypoosmotic challenge, to manage water loss and salt expulsion, aren't designed for the inverse problem. They can't reverse function on demand. The fundamental architecture of teleost osmoregulation is directional, and the fish can't simply reduce its TMAO to relieve the osmotic problem. Reducing TMAO means losing pressure protection. Proteins would begin to unfold. Enzymes would stop working. The fish can't trade the pressure problem for the osmotic problem. Both roads lead to death. The molecule that allows deep-sea fish to survive at all is the same molecule that ultimately makes survival impossible below a specific depth. A 2014 BBC report summarizing Yancey's work stated plainly that scientists considered it unlikely that any fish can survive in the oceans deeper than about 8,200 meters. This is a strongly supported hypothesis, a predictive model grounded in measured data, not a proven universal physical law. But the observational record aligns with it precisely. No confirmed teleost fish has ever been documented below approximately 8,200 to 8,400 meters. The Mariana snailfish at 8,178 meters sits right at the predicted edge. The famous flatfish that Walsh and Pickard reported seeing on the floor of Challenger Deep in 1960, near 10,900 meters? Marine biologists have since reviewed that claim and consider it almost certainly an error. The creature was almost certainly a sea cucumber, a soft-bodied echinoderm with no TMAO ceiling, misidentified in difficult visibility by two people who had never been to the bottom of the ocean before and had no frame of reference for what they were seeing. Fish at 11,000 meters don't exist as far as any evidence can determine. The TMAO ceiling predicts their absence. The absence confirms the prediction. Anthropods, by contrast, thrive in Challenger Deep at its full depth. Hirondelia gigas,
a scavenging crustacean, lives at 10,000 plus meters without issue. The reason is structural. Amphipods are osmoconformers. Their internal osmolality already matches seawater. Accumulating TMAO to handle pressure doesn't create the same osmotic crisis it does in teleosts, because there's no osmotic gradient to defend in the first place. Their body plan simply doesn't face the paradox that strands fish at 8,200 meters. This is why every survey of Challenger Deep's deepest sediments finds crustaceans and single-celled organisms, but no fish beyond a certain depth. The chemistry draws the line, the biology obeys. Size in the hadal zone follows the same logic. In the abyssal zone, where fish can actually survive, deep-sea gigantism is real for some taxa. Cold temperatures slow metabolism, reduced predation pressure allows longer lifespan, and extended growth periods produce unusual body size. But in the hadal zone, the TMAO constraint imposes an inverse selection pressure. A larger body carries more protein volume, which needs more TMAO to keep functional under pressure, which drives internal osmolality higher, faster. A fish optimized for the hadal zone needs to minimize structural cost. Smaller body, poorly mineralized skeleton, reduced muscle mass, gelatinous form that requires less protein per unit of volume. The trench doesn't produce giants among fish. It produces the biochemically leanest organism that can survive at all. This is the corrective to the mythology around the hadal zone. The deepest trench on Earth isn't the home of Leviathan-class organisms waiting to be discovered. It's the place where vertebrate life compresses to its minimum viable expression, where the chemistry of pressure imposes a hard ceiling on complexity and size, and where the only things surviving at full depth are creatures whose body plans happen to be exempt from the osmotic paradox. The trench isn't a refuge of giants. It's the end of gigantism. One thing that turned out to not be exempt from human industry. In February 2017, Alan Jameson at Newcastle University and his colleagues published a study in Nature, Ecology and Evolution documenting what they found when they analyzed amphipod crustaceans collected from within the Mariana Trench at depths between 7,841 and 10,250 meters. Every single sample contained polychlorinated biphenyls, PCBs, and polybrominated diphenyl ethers, PBDEs. Industrial chemicals manufactured widely from the 1930s through the 1970s and 1980s, when regulatory bans finally arrived, decades after their extreme environmental persistence and toxicity in biological tissue became impossible to ignore. The concentrations weren't trace readings in a pristine environment. PCB levels in the Mariana amphipods measured between 147 and 905 nanograms per gram of dry weight. Newcastle University's press release stated that the study had uncovered the first evidence that man-made pollutants have now reached the farthest corners of our oceans. But the comparison that makes the numbers visceral, PCB concentrations in these Challenger Deep amphipods were approximately 50 times higher than those found in crabs from the Liaohe River in China, one of the most heavily industrially polluted river systems in the world. The deepest point in the ocean was carrying more concentrated industrial contamination than one of the most contaminated rivers on land. A concurrent paper in Geochemical Perspectives Letters titled it plainly, "Toxic Anthropogenic Pollutants Reach the Deepest Ocean on Earth." The route these chemicals traveled follows the same physics that governs everything else in the trench. PCBs and similar persistent organic pollutants are hydrophobic. They bind to organic particles suspended in the water column. Marine snow, drifting down continuously from the sunlit ocean, collects these compounds. The particles sink. The V shape of the trench geometry funnels everything toward the deepest axis, concentrating it, the same physical effect that makes the hadal zone food-rich compared to surrounding abyssal plains. The design that concentrates organic nutrients concentrates industrial waste with identical efficiency. A compound manufactured in the Northern Hemisphere in 1965 and released into river runoff eventually enters the marine food chain, attaches to organic particles, sinks through 11,000 meters, and accumulates in the tissue of a crustacean at the bottom of the world.
Microplastics followed. A 2018 study documented microplastic particles specifically in sediments from Challenger Deep, not just the broader hadal zone, but the deepest point itself. Microplastic abundances in hadal bottom waters ranged from 2.06 to 13.51 pieces per litre, several times higher than in open ocean water. A plastic bag, the everyday grocery store variety, was found at approximately 10,975 meters depth. Researchers who examined the stomach contents of amphipods from Challenger Deep found synthetic particles in 100% of specimens. Every amphipod from the deepest place on Earth had ingested plastic. The Mariana Trench samples carried the highest mean ingested particle count of any location examined in the study. Victor Vescovo, during his record-setting dive to 10,928 meters on April 28, 2019, 4 hours on the floor of the eastern pool of Challenger Deep, reported seeing a plastic bag at the bottom. Don Walsh, who had first reached that floor 59 years earlier, was on the support ship when Vescovo came back up. The Mariana Trench formed because the Pacific Plate has been subducting beneath the Mariana Plate for tens of millions of years. Challenger Deep is a geological outcome of processes operating on timescales that reduce all of human history to a rounding error. The deepest point on Earth survived the formation of the Pacific seafloor, hundreds of millions of years of geological change, ice ages, mass extinctions, and the entire evolutionary history of vertebrate life. It took human industrial production approximately 80 years to contaminate it measurably, and about 50 years after that to put a plastic bag on its floor. The Mariana Trench isn't a mystery. It's a place where the physics of pressure rewrite the rules of biology, where fish can't survive below 8,200 meters, not because the depth crushes them, but because the chemistry of surviving pressure makes it impossible for their osmotic architecture to function. Where the hadal zone compresses vertebrate life to its biochemical minimum, rather than expanding it to mythological scale, where single-celled organisms outlast everything with a backbone, and where the funneling geometry that makes the trench biologically unique makes it simultaneously a terminal sink for the consequences of human manufacturing. The deepest point on Earth turned out to be close enough for that.